Okay, so thanks very much for inviting me to speak. I'm going to talk to you today some work that we haven't previously presented, looking at the role of CBP alpha leukemogenic alleles in our zebrafish model system. So just a little bit of background about CBP alpha. CBP alpha is a member of the BZIP transcription family of transcription factors, and it's essential for myeloid differentiation. Specifically, it's required for the differentiation of common myeloid progenitors to GMPs. It's also required for stem cell maintenance, and this is most marked during the fetal liver hematopoietic stem cell development. But also knockout of CBP alpha in adult hematopoietic stem cells results in a reversion back from quiescence to a more proliferative embryonic fetal stem cell type phenotype, suggesting that it maintains quiescence in adult hematopoiesis. CBP alpha is also involved in other tissues in adipogenesis and metabolism, and also lesser roles in T cell development. Structurally, the gene is on chromosome 19q in humans, and it arises from a single exon gene. There are two specific isoforms that are commonly expressed in hematopoietic tissue in humans, and these arise in frame with one another. A 42 kilodalton protein, which arises from nucleotide 150, and a 30 kilodalton protein that arises from nucleotide 504. These have 2 and 1 transactivation domains respectively, but they share this BZIP region, which is comprised of a DNA binding domain and a leucine zipper domain, which permits DNA binding. And a little bit more about the binding of the transcription factor. So CBP alpha requires dimerization in order to bind DNA, and this can be a homodimerization or a heterodimerization. Heterodimerization with CBP alpha has been described in detail for other CBP factors, including DDIT3 and CBP beta. But this data from Amy Keating's lab in 2013, modeling binding of the BZIP region of multiple human BZIP proteins, shows that in fact there's strong binding to a number of other BZIP transcription factors, including the ATF family and the BATF family. And this is important when we go on to talk about some of the leukemogenic mutations that we see in humans. So we're interested in CBP alpha because it's known to be mutated in human acute myeloid leukemia, and it occurs in around 10% of sporadic acute myeloid leukemia cases. These can be divided, broadly speaking, into cases that have a single mutation in CBP alpha, so a monoallelic mutation, and patients in whom there are 2 mutations, one on each of the alleles. Single allele mutations cluster around the N-terminal region of the protein, and these are generally out-of-frame mutations, which result in expression of a truncated protein of the P42, and so exclusive production really of P30 allele from that allele. When there's double mutations, we see quite a distinct pattern of mutations. So one of the mutations is usually in the N-terminal region, similar to what we see in most of the singles. And then the other mutation on the other allele is found in the DNA binding domain or leucine zipper, clustering just right around where these two things join. And these are in-frame insertions or deletions. And so they abrogate DNA binding and dimerization, but they don't result in loss of protein product, and they retain all of the other components of the protein. So just to recap that, when we look at all mutations across CBP alpha mutated AML, 95% of those with an N-terminal mutation will have an out-of-frame mutation resulting in P30 expression only, and 91% of C-terminal mutations will be one of those in-frame mutations that abrogates DNA binding or dimerization. So we're particularly interested in studying familial leukemias because importantly, in this situation, you know what the gene of onset, the mutation of onset is, because the families are born with it. So this is a summary of 10 families that had CBP alpha mutations, and each of these has an N-terminal mutation. resulting in the frame shift and production of P30 only. And interestingly, there's 1 or 2 families where there's incomplete penetrance, suggesting that there are genetic or epigenetic modifiers that are found in some patients that modify their disease phenotype. So just to recap, this is the wild type situation where there's P42 and P30. These are balanced in normal hematopoiesis. You can have an N-terminal mutation which truncates and gives you P30 only, or a C-terminal mutation which gives you aberrant binding. And quite a lot is known about these mutations in the chemogenesis from some elegant mice modelling done from Klaus Nerloff's group. And to summarise, what they have found is that C-terminal mutations appear to result in increased proliferation of early hematopoietic stem cells, and N-terminal mutations are permissive to some differentiation down the CMP to GMP pathway, and therefore the combination of expansion of the hematopoietic stem cell pool and some differentiation, which has now been shown to be requisite for the development of acute leukemia, have been defined as the mechanisms of leukemogenesis. However, we're interested in a number of questions, which is specifically, what are the effects of these mutations during development? Because patients have these in a number of situations during development. And secondly, what are the additional second hits and the role of additional second hits in addition to CBP alpha C and N-terminal mutations? So to address some of these questions, we developed a model of N-terminally mutated CBP alpha in the zebrafish. Zebrafish CBP alpha gene is highly conserved with human, and it exhibits the same isoform pattern with a couple of shorter in-frame isoforms as well. These are also seen in humans, but they're less well studied. And this is a transfection of the wild type or N-terminally mutated zebrafish gene into 293T cells, showing that you get the same kind of pattern of 2 dominant protein isoforms, the longer of which is lost when the N-terminal mutation is present. So we generated a TALEN-induced frameshift mutation at lysine 75, and this little red triangle here shows you where this is analogous to in the human protein. So initially we wanted to do a basic analysis to see whether this had any effect on hematopoiesis. And for those of you who don't know what zebrafish development looks like, this is a schematic of a 5-day-old zebrafish. This is its head, its body, and its tail. And hematopoietic cells arise along the dorsal aorta as they do in mammals. And these are here in the fish. And then cells are seeded from the dorsal aorta into the tail region. And this is usually around 2 to 3 days after they've been born. And this is termed the caudal hematopoietic tissue and is generally considered to be analogous to the fetal liver in mice. Once cells have proliferated and differentiated in the fetal liver, they then circulate again and seed into the definitive hematopoiesis organ in the zebrafish, which is the adult kidney. And this is rudiment of this starts around here. So this is an in-cross of N-terminal mutants, and we've done a Sudan black stain which looks at mature myeloid cells, and each of these little black dots that you can see in the tail of the fish is a single myeloid cell. So CBP alpha wild type and heterozygous animals have normal numbers of myeloid cells in this fetal liver region. However, CBP alpha N-terminal homozygotes have absolutely 0 mature myeloid cells at this time point. Growth and survival of C-terminal N-terminal mutants are also severely impaired, and we see that in this particular experiment all of the animals were dead by 35 days, but we tend to find that some of them do survive a little bit longer, so up to about 6 weeks. And this is a weight comparison at 6 weeks with the remaining fish that we had, and you can see that there is a marked reduction in the size and weight of CBP alpha N-terminal homozygous mutants. So in murine models of this N-terminal mutation, they observe a number of different patterns of changes in hematopoiesis. Specifically, they observe the absence of mature myeloid cells that we observe, and they observe that during early development, but once the animals get to about 9 months of age, they start to see a myeloid proliferation phenotype followed by frank evolution into leukemia. And so we looked over time to see what happened in our animals. This is flow cytometry plot of zebrafish kidney in an animal that expresses mCherry from the lysozyme C promoter, marking myeloid cells, and GFP from the CD41 promoter, which marks in the low expressing cells hematopoietic stem cells, and in the high expressing cells the platelet equivalent in zebrafish, which is a thrombocyte. So in the forward and side scatter plot, in the sibling animals, you can see that there is a group of cells here, which are the myeloid cells, and these backgated show expression of lysozyme C mCherry. In the N-terminal homozygous mutants, we don't see any cells in this skate at all, but if we wait till 6 weeks of age, most of the animals have started to again develop some expression of lysozyme C mCherry, and in fact, this is quite proliferative, and we see that the amount of lysozyme C mCherry expression far exceeds that of the wild types or the heterozygous animals. This also corresponds to a reduction in the number of myeloid cells seen in the peripheral blood, suggesting this is an aberrant proliferation of myeloid cells with a peripheral neutropenia. So as I said, one of the things that we set out to do was to look at second hit mutations, and we had proposed to look at some of these other interesting genes on this long list of mutations that are associated with CBP alpha mutations. However, we were specifically interested in cases that were familial, and in fact we discovered that absolutely every patient who has a familial N-terminal mutation has a C-terminal mutation. And this is quite interesting because it's not simply that they have one, some of them actually develop new leukemias with a second CBP alpha mutation. So this is a specific patient who starts out with a CBP alpha mutation in the C-terminal in this purple clone here, and the inside subclones with EZH2. And he achieves remission from his leukemia, and then he develops what's termed a relapse, but in fact it's a completely new leukemia with a different C-terminal CBP alpha mutation. He has some more chemotherapy and goes into remission, but he had some cells collected here when he was in remission, and he undergoes an autograft from that original remission phase. Obviously, we didn't know that he had a familial mutation at this time. And then he subsequently relapses with this original red clone here and a number of other new mutations. So basically, the acquisition of a C-terminal mutation is requisite for the development of AML in patients who have a familial mutation. And it also seems to be a strong driver for this. It's a small gene. This isn't a stochastic effect. This isn't statistically likely to happen because of the size of the gene, any more likely than it is to happen with one of the many, many much longer genes that we see in this second hit pool. So to faithfully model this, we went on to generate a mutation in the C-terminal of CBP alpha in zebrafish, and we generated a deletion of 4 amino acids in this region where there are many human mutations, which are shown in blue here. And we did the same basic assay to define the phenotype of these animals. And interestingly, we see a different phenotype with C-terminal mutants. In this instance, we see that there's an increase in the number of myeloid cells that are expressed in the C-terminal heterozygous animals. But in the homozygous animals, we see a marked reduction, but in contrast to what we saw in the N-terminal mutants, there are still some cells. So although there are virtually no cells, there aren't no cells. There are some cells that are differentiating into more mature myeloid cells in these animals. We also looked to see what happened to these animals in terms of leukemia development. So all of the double mutants or compound heterozygous mutants have impaired survival. So the blue line is the CC mutants, the red line is the NC mutants, and the purple line are the NN mutants. And a proportion of the animals over time develop widespread GFP fluorescence from the CD41 promoter, suggesting a proliferation of hematopoietic cells. And when we look at the blood from these animals, we can see numerous blast cells, and then this is a section showing that there's a infiltrate in the zebrafish kidney, some of which expresses myeloperoxidase, suggesting that these are acute myeloid leukemias in these animals. So we wanted to have a look at the effects of these mutations on development. And so we first started looking at primitive hematopoiesis. Primitive hematopoiesis starts around 10 hours post-fertilization in a developing zebrafish embryo and is characterized by monopotent myeloid macrophage cells and monopotent erythroid cells. So erythroid cells are unaffected by CBP alpha mutations. However, early development of myeloid cells marked at 22 hours post-fertilization by the transcription factor POO1 shows that there are marked effects on the production of POO1 expressing cells with C-terminal heterozygous causing an increase in the number of myeloid cells, homozygous mutants causing a reduction, and then in N-terminal mutants we see a reduction, although it's not significant, with a more marked reduction again in N-terminal homozygotes. And this is just a picture to show you what the animals look like at this time. We've had to manually count all of these cells because this is the animal lying on its back with a very large yolk sac, and each of the purple dots is an individual myeloid cell crawling over the yolk. In the compound heterozygotes, we also notice that there's a profound loss of myeloid cells at this point in development, but you'll notice that we lose the effect of the individual mutations, and this perplexed us a little bit, and we wondered whether we just had a clutch, for example, the first time that had a specific phenotype that we weren't able to replicate, but we are able to replicate this, but we also replicate this. And we think that the most likely reason for this is that these animals come from mixed parentage, so we don't know whether the mother is a C-terminal or an N-terminal, but it would be one or the other. And we think maternal effects from heterozygous mutations may be driving the reversion of some of these heterozygous phenotypes, which you'll see throughout what we find. So a few hours later, at 30 hours post-fertilization, most of these myeloid cells are now located in this tail region, which is called the posterior blood island during this point of development. And at this time point, some of these myeloid cells will still be primitive derived cells from the monopotent lineages. However, some cells arising in this region are also early definitive cells with bipotency, so either erythromyeloid cells or lymphomyeloid cells also arise in the same region. So we can't be certain the origin of these cells. However, we do continue to see that these are abnormal both in homozygous states, where we see a reduction in both N-terminal and C-terminal, and in the compound heterozygous state. We then wanted to go on and look at definitive hematopoiesis in these animals and look at specification and emergence of hematopoietic stem cells, which in zebrafish, like in mammals, occurs from the dorsal aorta. So this little stripe of purple that I've drawn some arrows on is the dorsal aorta in a zebrafish. And as you can see, there's no effect of any of the mutations, either heterozygous or homozygous, on the number or location of hematopoietic stem cells expressing RUNX1. We recapitulated these findings looking at CD41 low expression by flow cytometry, suggesting that our earliest markers of HSC specification and emergence are unchanged in these animals. We used a third marker for hematopoietic stem and progenitor cells, which is c-MYB, and we observed quite a different pattern here. So in c-MYB, with c-MYB expression, we observed that there is an increase in the number of MYB expressing cells in C-terminal heterozygous, with a corresponding opposite direction reduction in the number of hematopoietic stem cells expressing c-MYB in the N-terminal. And we see no effect in the homozygotes in this instant. In the compound heterozygotes, we see that the number of hematopoietic stem cells goes up. And these are the same sort of images that you looked at the last time. There's a little zebrafish in the corner so you can see which bit of it we're looking at. And I think what's interesting about these images, so you still see the same stripe here. There are a number of other cells on the yolk that we don't count because these are probably myeloid-derived cells. But much of the increase in expression or reduction in expression actually seems to arise from this tail region here. You can see it again here. And this is where stem cells seed once they have arisen from the dorsal aorta. So they circulate and then they seed and then they expand. So this is the very primitive fetal liver region. We wanted to see what happened to these cells over time, and so we looked later in development. So this is now 3 days post-fertilization, where all of these cells are now in this fetal liver equivalent region. And we see some differences to what we initially observed. So initially we observed that C-terminal heterozygous resulted in an increase in cells, and this is no longer the case, but homozygous animals now show a decrease. N-terminal heterozygotes, again, now have no phenotype, but homozygous animals show an increase. And the compound heterozygous animals show a reduction in the number of stem cells that we see in the tail marked by CMIB. These are just the images from the tail region, which, as you can see, show marked increase in the N homozygotes and marked reduction in the C-term. We observed these effects over time, so similar effects are observed at 4 days and also at 5 days, although we don't have this image for 5 days, but this seems to be persistent during this period of development. So we wanted to know what was actually driving, from a transcriptional perspective, these changes that were happening. So we undertook RNA-seq of stem progenitor cells at 5 days post-fertilization. And these are just volcano plots showing that a large number of genes were differentially expressed between the homozygous C and wild type, the homozygous N and wild type, and the compound heterozygous. However, we didn't observe lots of changes between the heterozygous animals. This is just a PCA showing that the heterozygous animals in fact cluster in between all of the wild type controls, but we see separation of each of the homozygous animals in the PCA plot. So the largest number of genes that were differentially expressed were observed in the NN group, and there was significant overlap in the other groups, but the most striking of this was in the upregulated genes in all of the genotypes. We did some gene set enrichment analysis and some clustering, and these showed that there was a number of groups that appeared to cluster together in terms of their phenotypic changes, and these include the CC and the NN mutations in upregulated genes, the CN and the CC in downregulated genes, and then genes that were downregulated in NN compared to wild type, but upregulated in CC compared to NN. So kind of following on from this and thinking about what we'd observed in heterozygous phenotypes that appeared to be in polar opposite directions between the C and the N heterozygous mutants, with a view that the presence of an N-terminal mutation drives the development of a C-terminal mutation to revert a phenotype that that needs to be corrected. We were interested whether there were any genes that were upregulated in the CC but downregulated in NN, or vice versa. So these are genes that are changed significantly but in opposing vectors. And we identified a small number of genes that fit this bill, and some of which look interesting for further study, and this includes this PTPRJ, which is a protein tyrosine phosphatase gene which has been implicated as a tumour suppressor in a number of solid tumours, and it's also been implicated in the development of ETV6 RUNX1 ALLs, as well as FLT3 mutated AML myeloproliferation. In addition, we're particularly interested in this subgroup of genes because this is the group of patients that, this is what humans get. More than 80% of patients get a C and an N. So whilst the CC causes leukemia in a fish and a mouse, that's all very interesting, but actually in patients, they predominantly, almost exclusively, get a C and an N. So we looked at whether there was anything specific about these C and N only gene alterations, and we see some interesting findings and upregulated pathways, including some T and B cell receptor signaling pathways and PI3 kinase pathways. And finally, we wondered if we looked again at these unique gene subsets, either in CC or in NN, whether that would also facilitate us, helping us to define what might be the selective pressure from a non-DNA binding mutant, for example, that might drive leukemogenesis or revert a phenotype from an NN. And this is the most interesting thing that we find, which is Genes unique to CC mutants that are upregulated are all genes that seem to be involved in cytokine signaling pathways, particularly IL-3, IL-5 and GM-CSF. And it's previously been shown that expression of these cytokines can actually override complete absence of CBP alpha and drive differentiation in aberrant hematopoietic stem cells, which leads to the development of leukemia. So in summary, we've developed zebrafish N and C terminal alleles that phenocopy human allelic mutations and result in AML. We've shown heterozygous and homozygous perturbation of CBP alpha differentially disrupts primitive myeloid development, and this is allele dependent and mutation site specific. We've shown that definitive myeloid primed HSPC development is disrupted temporally and in mutation specific patterns. And our gene expression studies show that CN compound heterozygous stem cells during early development show distinct transcriptional signatures, and that DNA binding null CBP alpha shows increased myeloid cytokine production upregulation. Finally, I'd just like to thank the people who did the work. This is largely the work of Catherine Hawkins, who's a clinical research fellow in my lab, with the informatics analysis done by Alex Lubin, with some assistance from Yvette Hode and other technicians in the lab and some of the alumni here. And I'd also like to thank the funders.